Hi guys, welcome to my all-in-one biology video, and this is for EDEXCEL GCSE 9 to 1. I hope you find it super helpful. As with all my other all-in-one videos, I try and go through every single specification point. All my perfect answers flash up, and don't forget, if you want to buy my revision guide, which I have written, containing my perfect answers, you can get that on the website, which will flash up now. I hope you find this video super helpful. I hope your studies are going well. Don't forget to come follow me on Insta, Twitter and Facebook, because I add lots of extra exam tips, extra explanations and just genuine cool science stuff, particularly on Insta. So I really would recommend giving that a go, and I do go through past papers there too. So yeah, I hope you find this video helpful. Let's get started. So first of all, our animal cell, remember it looks like a fried egg basically. It's got a big border and then it's got a small compartment in the middle. Remember that compartment is the nucleus. The nucleus controls the activities of the cell, so there's your key definition. Then you have some jelly which surrounds it, and we call that the cytoplasm. Role for that is that it's where all the chemical reactions take place, so that's another key definition. Surrounding the cell you have a cell membrane. Please note that there is no cell wall in an animal cell. The only thing you have is a border, a single border, which is the cell membrane, and that controls the entry and exit of substances inside the cell. Then you'll have to learn mitochondria. That's really the powerhouse of the cell, and it creates energy via aerobic respiration. Remember, aerobic respiration means that it requires oxygen. Then you have some tiny dots. These are ribosomes, and they're responsible for protein synthesis. Moving on to the plant cell, you find exactly the same structures in the plant cell. Nothing's missing, you've just got a few additions. And first of all, make sure you're happy that a plant cell is very rectangular in shape. And that's because surrounding it is the cell wall, and that supports the cell. Rectangle in the middle, called the vacuole, that contains cell sap. And then we've got a nucleus squeezed into one corner, again like the animal cell, that controls the activities of the cell. We also have mitochondria, but crucially we have some green things which look very similar to mitochondria, but the fact that they're green tells us that they're chloroplasts, and they're full of a pigment called chlorophyll, which gives it its green colour. That's where photosynthesis takes place. Bacteria are responsible for some of the most horrendous diseases out there, things like tuberculosis. They're bacterial diseases. Cholera is another one. This time you've got more of a distinct cell. You have a cell wall. You have a little tail, and we call that a flagella, and that helps the bacteria move to where it wants to go. Sometimes you'll see diagrams with a capsule around the edge or a slimy coat. That's just an additional structure. Don't worry too much about that. The crucial thing about a bacterial cell is that there's no distinct nucleus. What you have instead is a nucleoid, and then you'll see some tiny other structures, and these are plasmids, and these are also genetic information, and they're really important when we're talking about genetic engineering. Now, some of these adaptations, I think you can see they're not really distinctive adaptations. It's more about the fact that you're able to describe the cell in detail. So I do recommend that you do shove in as many specialist words as possible. Sperm cells now, I like sperm cells because they're really easy to remember. First of all, remember that they have a very characteristic shape. They have a whiplash tail, which helps them to swim. They have a central portion which is packed full of mitochondria to provide energy to enable them to swim. They have a head containing genetic information. And lastly, they have some enzymes in their head which help break down the outer casing on the egg when they need to penetrate it. Those enzymes are found in a special structure called the acrosome, another very good specialist word. So if I draw a very rough diagram of the female reproductive system, remember that the egg cells are produced in the ovary, and in terms of their specialisms, so how they're adapted for their function, you need to learn the following. They contain a haploid nucleus, which really means they contain only one set of chromosomes, and that will become much clearer later on in the video. The cell membrane, so this bit here, becomes hard after fertilisation, so once the sperm has fused. And why? Well, that's to prevent other sperm coming along and fertilising the egg. It has lots of cytoplasm, which we know is where chemical reactions take place, but the egg cell cytoplasm contains plenty of nutrients for the growth and development of the future embryo. And lastly, a jelly coat provides protection and again hardens after fertilisation.
Now we know that this egg gets deposited in the oviduct or the fallopian tube, and this point here that I've marked with an X is where fertilisation tends to take place at the entrance of the oviduct. So what is special about the cells which line the oviduct? Well, they have cilia. So if I zoom in on one, so these little cells line this oviduct, and we zoom in on them, and the cilia are really small hair-type structures, and they waft back and forwards, and in this way, when they waft together, they can actually move that egg along the oviduct. So that's how the cells lining the oviduct are specialised for their function. Linked with the cilia found in the oviduct are the cilia found in the trachea, which, as I've said here, the trachea is the windpipe. So you can see, here's our windpipe, our trachea, and these are our ciliated cells, complete with their small hairs. And why do we have them? Well, as we know, we breathe in through our nose, through our mouth, and that air passes down the trachea. However, it's not a perfect system, and sometimes pathogens and other particulates, dust for example, gets drawn in as well. Now we don't want those particulates ending up in our lungs. So the amazing thing is that there's mucus which traps the pathogens and the bacteria, and it causes it to stick to the cilia. And then, as I said previously, the cilia waft back and forwards, and in this way they can actually move the pathogens out of our trachea into our mouth, where it can be swallowed and destroyed by the hydrochloric acid found in our stomach. So let's just quickly make a note on the ciliated cells.
We're going to use the same thing in order to help you remember these definitions, because it's a hierarchy, they all build on each other. So remember, organelles are the small structures found inside cells. So an example could be mitochondria, it could be nucleus, it could be chloroplast. Now, a cell, in that case, therefore, is a group of organelles working together to perform the same function. And you're going to use those words to surround each of your answers. Now, if that's not making sense, I'll move on to the definition of a tissue. And you're going to say it's a group of cells working together to perform the same function. And then if you move on to an organ, well, it's a group of tissues working together to perform the same function. And then lastly, you've got an organ system, which, as you might expect, is a group of organs working together to perform the same function. So let's start by looking at the microscopes. Remember, there are two microscopes you need to know about. They are the light and electron microscope. Get those sorted in your head as to what they are. The light microscope you find in your classroom. You've obviously used it in science lessons, I hope. Electron microscopes are the ones you find in big scientific laboratories, and they are very expensive. So in terms of comparing how well they actually work, look at the magnifying powers. While light microscopes only magnify up to 2,000 times, electron microscopes up to 2 million times. Then we'll look at the resolving power, which is the ability to distinguish between two separate points. Light microscopes can only distinguish between points which are 200 nanometers apart, electron microscopes 0.2 nanometers apart. There are good things about light microscopes. You can use living samples. For electron microscopy, they very much have to be completely dead. And light microscopes you can see in colour. Electron microscopes, it will be black and white, but you often see pictures which have been coloured in or dyed. Now I'm going to show you how to use the magnification equation. This is your formula triangle that you need, which is image size, magnification and object size. So obviously if you're looking for magnification, you cover that. So you see that it's image divided by object size. If you're after image size, then you just do magnification times object size, and for object size you just do image divided by magnification, and I always use this triangle here to help me. Remember, when you're subbing in your numbers to the formula, you've got to use the same units, otherwise you'll screw up. So I recommend converting everything to millimeters. So to do centimeters to millimeters, you take your number that's been given in centimeters and you times it by 10. If you've been given the number in micrometers, then you want to divide it by 1,000. I'll show you an example now. So the image of a cell in a book is 48 centimeters in length. However, the real cell is only 120 micrometers. Calculate the magnification. So I'm going to use the triangles to tell me that magnification is image divided by object. Then we need to get the image size into millimeters. So I've been told it's 4.8 centimeters. So according to my
writing in black, I need to times it by 10 to get it into millimeters, so it becomes 48. And then we need to convert the 120 micrometers, which is fuzzy over here, into millimeters by dividing 120 by 1000, which is how I've got 0.12. Plug that into your calculator and you get an answer which is a magnification of 400 times. Let's look at another question. A student measured the diameter of a capillary in a micrograph. The image measures 5 millimeters and the student knows the magnification is times 1000. How many micrometers is the diameter of the capillary? Using our formula triangle then, we're after the object size, so we're going to do object size equals image divided by magnification. So I've written the equation here. And then we're just going to pop those numbers in as they are. So we're going to do 5 divided by 1000, which gives us a value of 0.005 millimeters. However, it wants the answer in micrometers, so therefore we need to do the opposite of my conversion. So we're going from millimeters to micrometers this time. So obviously we need to do the opposite of this, which is times by 1000. So that brings the answer to 5 micrometers. An enzyme is a biological catalyst. This means that it speeds up the rate of a chemical reaction without being used up. Now, an enzyme has a very special part on it called an active site, and that's the biologically active part of the molecule. And what happens is the substrate molecule binds to that active site. It forms an enzyme substrate complex, which then splits up to form the useful product that we're after. And we're going to talk now about digestive enzymes because I think it makes sense for us to look at the various products and substrates involved in digestion. So there are various enzymes you need to be aware of. Firstly, amylase, and notice the enzymes tend to end in ASE, so A's. Amylase is made in the salivary glands, in your small intestine and your pancreas. And what amylase does is it catalyzes the breakdown of starch into glucose. So in this case, starch is the substrate, it's the thing entering the enzyme, which is amylase, and then the product here is glucose. So what you can see is a very large sugar is broken down using amylase into a much smaller, simpler sugar called glucose. Now we need to look at protease. This is more straightforward because, as the name suggests, it breaks down proteins. So protein is the substrate and it breaks down proteins into amino acids. These are the products, and protease is found in your stomach, in your small intestine and your pancreas. The last enzyme you need to be aware of is lipase. Lipase breaks down lipids, or fats is their more colloquial name, and lipids are broken down into fatty acids and glycerol. So do try and remember that's the most complicated one. Now we need to just touch on enzyme activity. So the two things you can alter is both the temperature and the pH. So if you look at this graph, you can see at low temperatures, the enzyme activity is low. The reason for this is all to do with chemistry, it's to do with collision theory, because at low temperatures, enzymes have very little kinetic energy, as do the substrate molecules, so it means that the enzymes and the substrate aren't coming into contact very often. That means they obviously can't bind at the active site, and so the reaction can't be catalyzed. As we increase the temperature, you can see the enzyme activity increases. That's because those molecules are coming together more often, and at 37 degrees, as is the case with most animals, you will find that enzyme activity has reached its peak. It is at its optimum temperature, the best temperature, and that means the enzymes and the substrates are coming together very frequently. After this temperature and above this temperature, we see a massive decrease in enzyme activity, and that's because the enzyme has become denatured. Never say the word killed. They're not living, they can't be killed. You need to say they're denatured. And what that means is that the enzyme's active site has changed shape, meaning that the substrate can no longer fit. If we take a look at pH now, you can see a very distinctive graph shape here, and that's because enzymes have different optimum pHs. And if the pH is either too high or too low, around that optimum pH, you'll find that the enzyme denatures, which is why you have that cone shape. Because let's look at the enzyme which has an optimum pH of 7. If you go to 6.5 or 7.5, the enzyme will denature. And this is true for all enzymes. Some enzymes prefer different pHs to other ones. So proteases in the stomach, for example, because they're surrounded by hydrochloric acid, they'll have an optimum pH of approximately 3, whereas throughout the rest of the digestive system you find a slightly alkaline optimum pH, so of around 8, which is why you can't take stomach protease and put it in the small intestine and expect it to be okay.
So we're going to leap straight in with testing for glucose. Now you want to use Benedict's reagent here, and you're going to heat your sample with some Benedict's reagent in a water bath. If glucose is present, you will see a brick red colour appear. If it's not present, then you'll just see the blue, which is the Benedict's reagent, and that is testing for glucose. Testing for starch is simplicity itself. You simply add a drop of iodine. Now iodine will turn blue-black in the presence of starch. It will stay like a yucky brown colour if there's no starch there. To test for proteins, you're going to add burette reagent. I don't know if I'm pronouncing it right, but I'll spell it right now. And what will happen if you've got protein is you will see a lovely mauve purple colour. Where are you rushing off to, Lyra? And then lastly, testing for fats, you're going to use the emulsion test. So first of all, start by adding some ethanol to your sample, which is a type of alcohol. Then you'll add some water. If fat is present, you will see a milky white emulsion or suspension, and it will just go cloudy effectively. Now we're moving on to transport, so let's first of all touch on the 3 types of transport. So we're looking at diffusion, osmosis and active transport. So you need to know the definitions of these terms in great amount of detail. Now remember, with diffusion, it's the net movement of particles from an area of high concentration to an area of low concentration. So that's the reason why you spray perfume in one side of the room where there's a high concentration of perfume particles, and it moves across to the other side of the room where you can smell it. That is by diffusion. It is a passive process. It does not require energy. Osmosis is very similar to diffusion, however it involves the movement of water, which is why our definition this time is osmosis is the net movement of water from an area of high water potential to an area of low water potential across a partially permeable membrane. Now potential is just a really posh way of saying concentration, so somewhere where there's lots of water to somewhere where there's little water, and do add that it's across a partially permeable membrane. If there's no partially permeable membrane, it's not osmosis, it's just diffusion. So notice when water leaves the stomata from that leaf, that is by diffusion, because stomata is a whole, not a partially permeable membrane. Lastly, active transport. As the name suggests, it's an active process. This means it requires energy. The reason being is because it's the net movement from an area of low concentration to an area of high concentration, so against the concentration gradient. Let's touch on amoeba now. Remember, the amoeba is an example of a protoctis. This is a single-celled organism which can use diffusion in order to obtain all the nutrients it needs. So oxygen diffuses from outside that amoeba into the amoeba from an area of high concentration surrounding the amoeba to a low concentration inside the amoeba. The reason why diffusion is appropriate is because it occurs very quickly, because the amoeba is single-celled, which means it has a large surface area to volume ratio, and therefore the speed of diffusion is fast enough to allow oxygen in as and when it is required. Larger organisms, which are multicellular, have a much smaller surface area to volume ratio. Diffusion is not suitable, it is too slow, which is why they develop the need for a circulatory system. Now we need to talk about the cell cycle. So let's start by looking at chromosomes. So remember that your DNA is found inside chromosomes, which are found as pairs inside the nucleus of the cell. And this is why we say that the nucleus controls the activities of the cell. Now obviously, throughout our life, our cells don't just stay as old as we are, because you'd have an issue of some very old, worn out cells not doing their job properly. So obviously they need to copy themselves and produce daughter cells, i.e. new cells, so we can carry on living, and that's where the cell cycle comes in. Now remember that during the cell cycle, all that genetic information is copied, and that happens in mitosis, and then the cell divides in order to form 2 daughter cells, each with a full set of chromosomes. Now remember that in the cell cycle, in order to produce those genetically identical daughter cells, not only do we need to copy the genetic material, but we need to produce new organelles, so new mitochondria, new cytoplasm, new cell membranes, obviously, so that our daughter cell can be exactly the same as the original cell. Mitosis is the type of cell division which produces clones, so identical daughter cells, and these are needed in the growth and repair of organisms.
So mitosis, we know, is a type of cell division, and it's used for the growth and repair of cells. And in order to grow and repair, we obviously need to take our original cells and we need to multiply them. So we've gone from one cell, and we call this the parent cell, and we've produced two daughter cells. And remember, in mitosis, these will be genetically identical because they need to carry out the same roles. But there are various stages of mitosis you need to know. And I like to use PMAT to help me remember this. So P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. And you need a brief summary of what happens in each stage. And we know I can't draw, but what we're going to do is, here's my cell, and I'm going to say that the nucleus has broken down in it and spindle fibres form. So this is what happens in prophase. Then in metaphase, what you find is that the chromosomes line up at what's called the equator, so the middle part of the cell. In anaphase, the chromosomes separate and begin to be pulled to the opposite ends of the cell on these spindle fibres. And then with telophase, this is at the end of anaphase, and you find effectively that those two cells start to separate, two new cell membranes are formed, and you end up with two daughter cells. So I'm trying to show these separating, but I'm not doing a very good job. So your summary for telophase, a membrane forms around each set of chromosomes, and you obviously find those chromosomes in the nucleus. And new nuclei are formed. Tiny side point, cytokinesis. This is when our new cells, which are diploid because they contain two sets of chromosomes, are separated by new cell membrane. So what is growth? And there's quite a few little definitions you need to learn. So first of all, the obvious one, it's an increase in size of a particular organism. It can be an increase in the number of cells. An increase in the size of those cells. So we go from a small cell to a big cell. In the previous, we go from 2 cells to 4 cells. And then lastly, how can you measure this growth? Well, you can measure it by comparing the new mass or the new length.
We're going to talk about stem cells and differentiation. Now remember, a stem cell is a cell which is capable of becoming any other type of cell. So they're not specialised, and that's why stem cell research is so important now, because if you could harness a stem cell and get it to produce a hair cell, an eye cell, a skin cell as needed, then you'd have a very powerful tool. Now in animals, cells become specialised at a very early stage of their development, so when they're an embryo, for example, and it is very difficult. You can't actually get adult cells to become any other type of cell. Plants are different to animal cells because they have stem cells which can constantly differentiate throughout their lives, and that's great for the plant because at any one point these cells can become any type of plant cell that the plant requires. These growing areas where mitosis occurs in a plant are found in meristems, which you find in the shoots and root tips.
So what are meristems? Well, first of all, they're a group of cells, and crucially they're found in plants at the end of shoots and roots. And the purpose of meristems is that they allow plants to grow continually throughout their life. And this growth is obviously due to a type of cell division, which we've already met, which is mitosis. And the cells either grow in number or they differentiate and become other cell types, depending on the need of the plant. And this bit here is key, the fact that the meristems can differentiate into any other type of cell found in a plant, because this isn't really true for us humans or other animals. Our cells tend to have their own function unless they're particular stem cells. So a bone cell will always, when mitosis occurs, will become a bone cell, the same with a skin cell, cheek cell, etc. So the brain, I'll try and find you a good picture of the brain because I honestly cannot draw to save my life, but here's a very rough picture. And let's give the brain some very basic labels. So the top part of the brain, the bit that looks like a brain, is known as the cerebral cortex. This bit down here, it looks a bit like a cabbage in some diagrams, is the cerebellum. And then underneath here you have the medulla and the brainstem. In terms of adding a bit more detail then, so I've already said cerebral cortex is the bit which looks mostly like your brain or how you imagine your brain to be, so we're going to call this the main part of the brain. It's divided into two hemispheres, so half spheres. And in terms of its function, it's all to do with our emotions, our memory, what makes us us, so our personality, our behaviours. In terms of the cerebellum now, this is all to do with our balance, maintaining our posture. So if we have anything wrong with our cerebellum, we will struggle to stay on our feet. And now lastly, the medulla, or the medulla oblongata, if we're giving it its full name. This is really the control center of the brain. So it controls everything that keeps you alive. So it controls your breathing rate and your heart rate, so the speed at which both of these things happen. And lastly, your reflexes. So how can we actually look at our brains, look at its activity? Well, the most obvious way is through surgery, and electrodes are used to stimulate the brain. We can also do a number of different types of scan, which we need to look at in more detail. In terms of the advantage of using scans versus surgery to examine the brain, first of all, you get a more in-depth picture of the brain and you have very little risk of damage to the brain. So how can the health of our brain be determined? Well, there are several different ways. Firstly, using brain surgery. And here electrodes are used to explore the function of various parts of the brain. So you electrically stimulate parts of the brain, you keep the person conscious because there are no pain receptors within the brain, and you ask them how they're feeling. So for example, if you stimulate a portion of the brain that causes them to feel hungry, then you can be pretty much sure that the portion you're looking at is to do with feelings of hunger. But simply put, electrodes are used to explore function. And then secondly, you've got a wide variety of scans. So let's start with a CT scan. A CT scan uses X-rays, and these pass around the body. X-ray detectors measure the absorption of the X-rays. And then a computer builds an image of the structures found within the brain. A PET scan is slightly different. This time it shows the brain's activity. And in this case, radioactive glucose is injected into the person. And active cells, these are most likely to be cancerous cells, absorb this radioactive glucose. These cells then produce gamma rays which can be detected, and in this way the cancer can be located.
Now we're going to talk about nerve cells, and I'll try and add a picture of a nerve cell because they have a very unique shape. Now, first of all, they have lots of dendrites, and that allows them to make lots of connections with other cells. They have a very long axon, which is the central kind of actual nerve that forms the length of the neuron. And what that does is that it carries the nerve impulse from one place to another. Because they're very active cells, obviously they need lots of energy, so you'll find that they have lots of mitochondria to produce that energy in respiration. And lastly, the nerve endings have special synapses, which are gaps where a chemical diffuses across in order to convert that electrical impulse into a chemical one.
Let's have a look at this simple diagram of a sensory neuron now, and the bit I'm really interested relates to here, which is the axon. Now, surrounding the axon you have what's called a myelin sheath. And this is a fatty substance which acts to insulate the axon. Why does it need to insulate the axon? Well, it's to increase the speed of the electrical impulse transmission.
So we're going to take the nervous system in greater detail now. So let's first of all look at what a stimulus is. So that's the change in the environment, and obviously that causes the response that the nervous system brings about. Do be aware of what the central nervous system is. That consists of both the brain and the spinal cord. Let's go through all the steps involved in a regular nervous response, one which does not involve a reflex action. So I'm going to use picking up a book as an example. So first of all, we need to list the stimulus, which is seeing or viewing the book. This is picked up by receptors, and these receptors will be in your eyes, your photoreceptors on your retina. They'll send electrical impulses along your sensory neuron to your central nervous system. Electrical impulses then pass along your motor neuron to your effector, and this will be muscles or glands, and in this particular case it will be muscles which will contract to pick up your book. Remember, an effector is either a muscle or a gland, so a muscle responds by contracting, a gland responds by secreting hormones. Don't forget the role of the synapse. So the synapse is the gap between two neurons, and this is where a neurotransmitter is released. So the neurotransmitter diffuses across that synaptic gap, that synaptic cleft, and binds to the postsynaptic membrane. So your electrical impulse is changed to a chemical or a neurotransmitter and then changed back to an electrical impulse at a synapse. Looking now at reflex actions, remember these are faster and they are involuntary, so they do not involve a conscious part of your brain, and it tends to be in response to something painful. So putting your hand in the oven and accidentally touching one of the shelves, which is hot, this would trigger a reflex action. So the stimulus this time would be the high temperature from the oven tray. Your receptors would be on your fingers, which would receive that information about it being too hot. Electrical impulses would then flow along your sensory neuron to your relay neuron this time, so we're not involving the conscious part of our brains. The electrical impulse passes along the motor neuron to your effector, which will be a muscle in your finger, which contracts to remove your hand or your finger away from that heat source.
Now, our central nervous system is made up of both the brain and the spinal cord. And we've looked in some detail at the brain, now we need to look at the spinal cord. Now this travels down the length of your back. It's held in position and protected by the spine, which is the bony cage that protects it. Now, if you have damage to your spinal cord, clearly electrical impulses won't be able to reach to the bottom part of your body, so you end up with a loss of feeling in your legs. If you have damage higher up, so near your neck area, then you're going to lose that sense of feeling all the way down your body. So you're going to end up with loss of use of both your arms and legs, and we call this quadriplegia. So what sort of treatment options exist in order to repair damage to the spinal cord? Now, unfortunately, we can't use stem cells to treat spinal cord damage. And the reason being is that adult stem cells are unable to specialise or differentiate into other cell types. So regardless of the fact we need new spinal cord cells, for example, we don't have adult stem cells which exist that can do this for us. So let's just say NB, we're unable to use adult stem cells to repair the spinal cord. Because adult stem cells can't differentiate into any other cell types. So what treatment options actually exist? Well, you could use, if you can't use adult stem cells, you can use embryonic stem cells, because remember, in embryos their stem cells can differentiate into any other cell type. The only problem with this is ethical issues concerning the fact that you need to use embryos, so that is aborted or miscarried fetuses. So clearly we have an ethical issue here, but theoretically they could be used. And secondly, electrical stimulation. Now looking at treating brain tumours, here you can use a combination of radiotherapy and chemotherapy. Radiotherapy is when you use X-rays directed at the cancer to destroy the cells. With chemotherapy you're using chemicals, hence chemo, and these chemicals are drugs which kill the cells. But whichever way you use, you want to minimise the amount of damage you do to the healthy cells. So you want to make sure those X-rays really only affect the cancerous cells, and the same with the drugs. You don't want them going off and damaging healthy cells, because that's why people feel really sick on chemotherapy. There are several risks associated with treating brain tumours, and not just due to the things I've just talked about in terms of damage to healthy cells. You've got some major issues with the blood-brain barrier, which, as the name suggests, is the separation between the blood and the rest of the brain. And these drugs can readily cross the blood-brain barrier and can cause effects far wider reaching than just the brain.
Right, we're going to look now at the eye. You need to know lots about this. It's a very important sense organ. So I'm going to start by running through its various structures and their roles. So first of all, light enters the eye and it hits the cornea. So the cornea's role is to refract the light, to bend that light as it enters the eye. The light then has to pass through the pupil. So the pupil's role is simply to allow light into the eye. The size of the pupil, however, is controlled by the iris, and that is the colourful part of your eye. Mine's brown, other people's are green or blue. And the iris has circular and radial muscles which will actually help it control the size of the pupil. We'll go into that in slightly more detail further on. So now light has entered the eye, it now hits the lens. The lens's role is to refract that light further in order to focus it on the retina. The retina contains photoreceptors. These are cells which are sensitive to light, and they are called rods and cones. Rods are sensitive in very dim light, and cones are sensitive to colour. Then we have the optic nerve, and that converts those light signals into electrical impulses which can be carried to the brain where they can be computed. The white bit of your eye is the sclera, and that just forms the tough outer casing. If you've ever dissected an eye, you'll notice it's full of some black inky stuff. That is the choroid layer, and that's role is to prevent light bouncing around within your eye. Then we have a blind spot. The blind spot is simply the place on the back of the eye where the optic nerve leaves the eye, because clearly there can't be any photoreceptors where you have the optic nerve. The fovea is a place that is very concentrated with cones, so it's very good for seeing colour. We need to look a bit more now at the lens. So I've already told you that the lens focuses light onto the retina. It does this by a process of accommodation. So accommodation is all about focusing light from different distances away so that an image can be formed on the retina. So let's look at what happens when we're looking at an object far away. So when it's far away, the light is going to be coming into your eyes fairly parallel, which means it doesn't need refracting very much. Therefore, the lens needs to be thin. In order to do this, the ciliary muscles relax and the suspensory ligaments are taut, and this means you have a very nice thin lens, so it doesn't refract the light too much. Looking at an object really close to us now, the light rays are going to be coming in at a far greater angle. Therefore, the lens needs to do far more work in order to focus light onto the retina. In order to do this, the lens needs to be fat. So in this case, our ciliary muscles contract and our suspensory ligaments slacken off, meaning that our lens is nice and fat. So that's the way in which we focus on light at different distances away. We now need to look at the pupil reflex. So the pupil changes its diameter dependent on light levels.
So if there's lots of light, the pupil needs to be narrower, and that's to prevent damage to the retina, because too much light entering the eye will damage the retina. If we're in a dim or dark room, the pupil obviously needs to be nice and wide in order to allow as much light into the eye as possible. We can use this response as an example of a reflex action. So previously I told you all the steps involved in a reflex action, and we're just going to use those steps, but we're going to apply them specifically to this example. So I'm going to use an example which is that we have walked into a very bright room. So the stimulus will be lots of light. The receptor will be the rods and cones on your retina. Then the sensory neuron will pass along the optic nerve to the brain, and there will be a relay neuron. Then we have a motor neuron, which is also passing along the optic nerve, and it will end in an effector. And in this situation, the effector is the muscles which you find in your iris, so your circular and radial muscles. And you'll find that your circular muscles will contract and your radial muscles will relax, and that will act to narrow or constrict the pupil. So now let's look at why some people need to wear glasses. And it's all to do with how the rays enter your eye and where they end up. Now the whole point is they're supposed to meet here, which is on the retina. But in this case of this person, you can see that the rays meet far too early. So the rays converge too early. Converge means meet. And so what's wrong with this person's eye? Well, their eyeball could be too long. As you can see, there's all this extra eyeball here. If it was a bit shorter, if you brought the eyeball into here, then there wouldn't be a problem. Or it could be a matter to do with the lens. The lens could be too strong. So remember, the role of the lens is to refract the light, and if the lens is doing that too well, it will do it too soon, as is the case here. And this person therefore is short-sighted, which means they can't see distant objects. In fact, that's exactly what I have. In order to rectify this, you can see the sort of lens we've got here. It's a concave lens, which causes the rays to spread out here. And so that when the lens does its job, the rays are brought sharply into focus on the retina. Rays now converge on the retina. Having a look at a slightly different issue, this usually affects older people. It's why you need reading glasses, because you can't see objects close to. So these people are long-sighted, so can't see objects close to or nearby. In this case, you can see that the rays converge behind the eyeball or behind the retina. So why could that be? Well, it could be that the eyeball is too short. It's almost like you need the eyeball to come all the way back here. But that doesn't tend to usually be the problem. It's usually that the lens is too weak, because over time the lens weakens and can't refract the light so well. And in this case, you need what's called a convex or converging lens. And as the name suggests, it causes the rays to come together exactly where they need to be on the retina. So look at the different types of lenses we have. The concave lens helps to diverge the rays, bring them out. The convex lens helps to bring the rays in. So in terms of correcting short-sightedness and long-sightedness, obviously we can see here you use lenses in glasses or contact lenses. The second thing you can do is laser eye surgery. And how that works is it kind of shaves portions off your cornea here, because if you have a look, the cornea is where the light first is refracted. So if you can actually shave the cornea, then you can help to fix eye problems here. Let's just touch on cataracts now, and we might as well use these diagrams here. So it's when the lens, so the bit that I'm highlighting, the lens becomes cloudy with cataracts. And obviously if it's cloudy, you're really going to pick up a very poor image. And the reason it becomes cloudy is due to protein. And the way in which you fix cataracts is by replacing the lens with a plastic lens. Let's look at mitosis and meiosis now. So remember, they're both types of cell division, but they're used for making very different things. So meiosis is used to make gametes, so that means it's used to make sperms and egg. Mitosis is a completely different type of cell division. You need to learn that it's used in cloning, asexual reproduction, and the growth and repair of cells. So for example, if you damage yourself, you cut yourself, it will be mitotic cell division, which replaces those cells. If you're carrying out asexual reproduction, so something like a strawberry runner producing baby strawberry plants, that will involve mitosis. The reason being is that it creates genetically identical offspring. Let's look at the differences between mitosis and meiosis. I always do this as a table because it allows me to make a direct comparison. So look at the number of cell divisions first of all. That will be 1 cell division in mitosis, 2 cell divisions in meiosis. The number of daughter cells now, so that's the number of cells produced once this cell division has taken place. In mitosis, you're looking at 2 daughter cells. In meiosis, you're looking at 4 daughter cells. I've already touched on the sorts of cells that are produced, but just to recap, mitosis produces genetically identical daughter cells. Meiosis produces genetically varied daughter cells, which makes sense. If we're using mitosis in cloning, that's genetically identical. If we're using meiosis in making gametes, it makes sense that we want our sperms and egg to all be different to each other. And do notice that the gametes will contain a haploid number of chromosomes, whereas the daughter cells produced by mitosis will contain a diploid number. Don't forget that haploid means containing 1 set of chromosomes, so in humans that is 23. Diploid means containing 2 sets of chromosomes, in humans that is 46. Looking at species now, so what is the definition of a species? Well, it's individuals which can reproduce to produce fertile offspring, and that's key. It's all very well having a horse and a donkey and they mate and they produce an offspring, which we call a mule, but the mule is sterile, it cannot reproduce. So that's the crucial thing about members of the same species, they can reproduce together to produce fertile offspring. Lyra! Hey! Did you come in? Oh, you're all soggy. Where's it? Where isn't it focusing? You're so soggy. Is it wet out there? Super soggy. So how is variation within a species brought about? Because we know the human race isn't full of billions of people that all look the same. That is brought about by a combination of things. First of all, genetics, and secondly, environmental factors. So 2 identical twins, regardless of the fact they have the same genes, if you move them to opposite parts of the world, it's very likely they'll have different heights, different masses, slightly different skin colour, and that's due to the environment they experience. That could be lots of sun, one of them could eat more, one of them could do less exercise. You're right. Are you too muddy? You're purring, aren't you? This makes a change. What is a mutation? It's a rare random change to the genetic material of an organism. So a mutation can be brought about by a number of things. Things like ionizing radiation, exposure to UV light, X-ray exposure, and various mutagens, which are just chemicals which cause mutations, and you find those in things like cigarette smoke. Now, the crucial thing with mutations is what they do is they alter the DNA of an organism. We've already looked at protein synthesis, so if you think about it, if you alter the DNA of an organism, what that will do is it potentially alters the sequence in which amino acids are assembled, and therefore it can alter the end product, so the end protein which is produced. Proteins are responsible for phenotypes, so our physical appearance, so mutation can therefore cause an alteration in our phenotypes. Now, not all mutations cause this alteration, because sometimes a mutation occurs where the DNA, although it is changed, it doesn't actually alter the order in which the amino acids are assembled. So you end up with the same protein here. Let's look at continuous and discontinuous variation now, and hopefully you've met these 2 words in maths, so you already have a clue a little bit about what they mean. Now, continuous is all to do with having a range of values, and it results in a range of phenotypes which fall between two extremes. So for example, your height. So taking people in your year, you'll have the shortest person in the year, which let's pretend they are 120 centimetres tall. The tallest person might be 200 centimetres tall. I don't know if I'm making up silly numbers, by the way. In the UK, for some reason, we described height in terms of feet, so I'm 5 foot 4 inches, which is really silly because we use the metric system for everything else. Whereas discontinuous variation results in limited phenotypes with no intermediate values. And we've already met a really good example, which is blood groups. You're only one type of blood. So you could be A, you could be B, you could be A, B or O, but you're not a combination of all four. The same could be said with something as silly as tongue rolling. The general thinking here is that some people can roll their tongues, some people can't, and you can't kind of do it halfway. And be aware that this is all to do with genes, which you should know from doing the genetic cross. Moving on to the protein synthesis part of the specification, we need to start by looking at some key definitions such as genome, and that is the entire DNA belonging to an organism. So we're focusing on the nucleus of a cell. Remember that the role of the nucleus is to control the activities of the cell, and it does this because it contains lots of genetic material. So within the nucleus we know that there are chromosomes. There are 46 chromosomes, which is a diploid number because remember they're arranged as 23 pairs. Remember, half the number of chromosomes is known as a haploid number. However, I digress. I don't really want to talk about that now. So the chromosomes are made up of DNA. You need to know the definition of a gene. A gene is a section of DNA which codes for a particular protein. Looking more closely at DNA now, so we need to first of all know what the structure of DNA is. Remember, it looks like a ladder. We call this a double helix, which winds itself up. It's made up of a sugar phosphate backbone, and remember that the sugar here is deoxyribose. Why? Because DNA stands for deoxyribose nucleic acid, so the sugar here is deoxyribose. There's a phosphate and sugar backbone, and then linking the two backbones, the rungs of the ladder, are bases. And remember the names of these bases: adenine, thymine, guanine and cytosine. Looking more in depth at the various names involved, nucleotide. A nucleotide is simply just 3 units made up of a deoxyribose sugar, a phosphate and lastly a base, so it could be either one of those bases I've just mentioned, so adenine, thymine, cytosine or guanine.
Sorry, is that how you pronounce it, guanine?
How do you say
I want to say guanine.
I got taught guanine.
Really?
But it sounds weird now you're saying it. Martin's telling me that I'm pronouncing guanine wrong. My biology teacher said guanine, and I don't know how to say it any other way. Apparently he says guanine. So, you're like, what an idiot. So we need to now look at complementary base pairing. That's when the various bases pair up in a very specific way. Try and remember the shape of the letters match. So guanine or guanine always pairs with cytosine because they're both curly in terms of the G and the C. Adenine and thymine always pair up, and that's because they're the straight letters. So use that to help you remember. Looking at the difference between RNA and DNA, the main difference here is the sugar involved. So RNA has ribose sugar as opposed to DNA's deoxyribose. And lastly, there's a change in base. There's no thymine in RNA. You've got to learn that this time it's uracil. So adenine pairs with uracil in RNA, and cytosine and guanine still pair up. RNA is single-stranded compared with DNA's double strand, so we don't see a ladder with RNA, we just see one side of the ladder effectively. Let's move on to looking what a codon and anticodon is. So a codon is just a fancy way of describing 3 bases which are found on the mRNA molecule, and they correspond to a single amino acid. An anticodon is the 3 complementary bases which you find on tRNA, and they pair up with the codon. So although I've touched on mRNA, tRNA, codons and anticodons, we need to actually understand why we're even mentioning these words. Hopefully you'll know this from your school classes. After all, this is a revision video, but now I'm going to go into great detail about protein synthesis.
So remember, protein synthesis is all about making new proteins. It's about sorting out the arrangement, the sequence of amino acids so they can actually produce a particular protein. And we're going to talk about how those amino acids line themselves up in the correct order, aka protein synthesis. So there are two main stages, transcription and translation. And the first stage is transcription, so we need to go into that now. So, bye cat. So with transcription, first of all, the DNA needs to unwind to expose a single strand. So we're going to expose those bases on the DNA. There are bases floating around inside the nucleus, mRNA bases, and they come along and they pair up with those exposed DNA bases. And that will start forming the mRNA molecule, and it's a single strand, noticing that uracil has replaced thymine. So we call this complementary base pairing, and a codon is simply 3 of those mRNA bases in a line. So we have created our mRNA, our messenger RNA, because it carries the message from the DNA, and now needs to leave the nucleus, so it leaves via a nuclear pore and attaches itself to a ribosome, which we find within the cytoplasm. Hence, the summary definition of a ribosome's role is that it carries out protein synthesis. So the mRNA attaches to a ribosome, and this is where translation begins. So there are tRNA molecules within the cytoplasm, and they have complementary anticodons. So they'll have the 3 bases which complementary pair up with the mRNA exposed bases. And at the other end of that tRNA molecule will be an amino acid, and effectively the tRNA brings that amino acid to the mRNA. The codon will match up with the anticodon, and we have our first amino acid in place. Then the next 3 bases on the mRNA will be read, and a different tRNA molecule will bring probably a different amino acid along, because if there are different bases, it will correspond to a different amino acid. So the second amino acid has been brought, and they attach to each other using a peptide bond. So that is the start of our protein chain. It keeps going, and eventually a stop codon will be reached, and that's just 3 specific bases which correspond to no amino acid whatsoever, and that will signal the end of that growing protein chain. You know the definition of a mutation, that's a random change in the DNA of an organism. So effectively those bases, we know that they occur in a certain order within DNA. A mutation will cause a change to those bases. So first of all, it could just be a straight substitution. It could be a deletion, so a whole base could just be taken out. And you find with a deletion is that effectively all those bases move along one place, so you'll end up with totally different amino acids being produced. It's quite likely that you'll end up with huge disruption in the protein made. There might be an inversion, which is when the 2 bases in the DNA swap over. There could be a duplication when a base is simply copied, so there could be 2 adenines in a row, for example, and again that will most likely cause a change in the amino acid which is brought along to the ribosome. We're going to talk about genetics now, so we need to know the definitions of lots of very key important terms, and then I'm going to show you how to do Punnett squares and pedigree analysis. So let's start by looking at what a gene is. A gene is a section of DNA which codes for a particular protein. Now there are different genes which control different traits, so for example eye colour. Now different forms of the same gene we call alleles, so you must learn that definition. So if we take eye colour for example, different alleles for eye colour could be blue or brown. You must now know the meaning of the word genotype. The genotype is the alleles an organism has. So for example, when we're talking about blue eyes, it's 2 small b's. If we're talking about brown eyes, it could be big B, little b. So when you're asked for genotype, you must provide letters. The phenotype is different. This is the physical appearance of a particular trait. So if you're asked for the phenotype of this eye colour, the answer here is blue. So the genotype would be little b, the phenotype would be blue eyes. So be very aware of that distinction. Next up, we need to know the meanings of homozygous and heterozygous. Homo means same, so it means having 2 of the same alleles, whether that's 2 big B's or 2 little b's, it doesn't matter as long as they have the same case. So they both need to be upper or they both need to be lowercase. And that is the meaning of the word homozygous.
Heterozygous means different, so that means containing different alleles. So in the case of eye colour, that would be a big B and a little b. Now, a dominant trait requires simply the presence of one allele for it to exhibit itself in an individual. So brown is an example of a dominant trait because you can have two big Bs or a big B and a little b and the trait will still appear. Recessive requires the absence of the dominant allele, so a recessive trait could be blue eyes because you need two little b's, for example.
A blue-eyed mother and heterozygous brown-eyed father decide to try for children. What is the probability that the children will have blue eyes? So let's work out what we have here first of all. Blue-eyed, remember blue is a recessive trait, which means that her genotype must be small b. We've been told that the father is brown-eyed, which means he could be big B small b or big B, but the fact that he is heterozygous tells us that he must be big B small b and not big B. So I'll show you how to lay out your answer. This is the method you should always use. So start by writing mother and father at the top. And we know how much I love tables. So you're going to write in your table phenotype, genotype, and lastly gametes. Remember these are sperm and eggs. So the phenotype, this is the physical appearance. We can see from the description that the mother has blue eyes and the father has brown eyes. The genotype, so these are the alleles that each parent has. I've already written these out, so it's small b, big B, little b. The gametes, just split these up because this is saying what the eggs and sperm will be. So just write out what you wrote on the genotype layer, but put circles around them to show that they're gametes, because here are the sperm and the mothers are eggs. And now we need to do the Punnett square. So mother, father. I don't know why my iPad sometimes undoes what I've done already. So she's small b, he's big B, little b. Let's cross them. So big B, big B, small b, small b. And therefore both of these will have blue eyes. Both of these will have brown eyes. So as a percentage likelihood, it's 50% will have blue eyes and 50% will have brown eyes. Cystic fibrosis is a recessive disease. A carrier mother and a carrier father decide to try for children. What is the probability that their child will have cystic fibrosis? So it's recessive, which means to have the disease you need this genotype, small c. It doesn't matter what letter you use to assign, by the way, but I'm using C here because of cystic fibrosis. A carrier mother and a carrier father, that automatically tells me that this is their genotype, and you must learn that they're effectively heterozygous if they are carriers. So we've worked out their genotypes, so we're ready to do the genetic cross by writing mother and father at the top again. Phenotype, genotype, and gametes in the table. The phenotype is that they are both carriers. Their genotype we know is heterozygous, so it's big C, small c. This means that half of her eggs will be big C, half of them will be small c, and the same with the sperm. Make sure you really show a difference in the size of your letters here. And now it's time to do the Punnett square. So this child is big C, so they'll be healthy. This child is big C, small c, so they'll be carriers, but they'll still be healthy. Same for this one. And lastly, this child here will have cystic fibrosis. So they have a 25% chance of having a child with CF. So let's now look at how we inherit our sex. And remember, this is all to do with chromosomes. So women have 2 X's and men have an X and a Y. And that is the pair of chromosomes which dictate your sex. So let's literally have a quick look as to why 50% of the population, or approximately 50%, are male, 50% are female. So let's start by writing mum and dad. As per usual, we're going to write phenotype. So what do they look like? Well, the mum's obviously female, the father is male. The genotype, you're going to use the sex chromosomes here, so she'll be XX, he'll be XY. The gametes, so obviously every egg will be X. Sperm, weirdly, can either be female or male sperm. And then if we look at the cross, let's cross the mum up here, the dad down here, and you see this situation which shows 50% are XX, so they're female, 50% are XY, so they're male. Although we know, based on that evidence, that the man's sperm determines the sex of their child, and the chromosomes, so the XX or the XY, contain lots of genes associated with the development of sex organs. You might not know that they have lots of other roles which might not be to do with sex at all, and they code for characteristics which are completely unrelated to sex. So the sex chromosomes also carry genes that code for characteristics unrelated to sex. And a very famous example of this is colour blindness. So that's whether you can see in full colour or not. And because it's carried on the X chromosome, so a sex chromosome, we say weirdly that colour blindness is sex linked. Let's look more closely at colour blindness as a sex linked characteristics, and a very well known one is red-green colour blindness, which is when you can't tell the difference between the colour red and the colour green. And it's carried on the X chromosome only. And when we do our crosses, just so you know, in order to have colour blindness, you need to have a small c. So if you're a guy, to have colour blindness, you need to have a X small c. The Y, you're not going to have a C on it, because remember I already told you that it's carried by the X chromosome only. As a female, to have colour blindness, you need to have 2 small c's. If you have a big C and a little c, then you're just going to be a carrier, and if you have 2 big C's, then you're going to be completely normal. So let's do a question to try and make this a bit more straightforward. So we're going to cross a carrier woman and normal man. So using our normal way of writing out our crosses, so we've got a woman and a man. The phenotype, first of all, well, we know the woman is a carrier, the man is normal. The genotype, as you look at my crosses up here, she needs to have a small c and a big C in order to be a carrier. And he's normal, which means he must have a big C attached to the X. And remember, the Y chromosome doesn't carry any genes for colour blindness, so we leave that as it is. So the gametes. The woman's eggs will either be big C or small c, and the man's sperm will either be big C or just a Y, which will obviously code for a male sperm. So now let's do the cross. So this first offspring will be just a normal offspring. The second offspring will be a carrier of colour blindness. This male offspring will be normal, and this final male offspring will have colour blindness. So let's write them out. 25% of the offspring will be male and have colour blindness, and that's due to their combination of alleles, which is X small c Y. 25% will be male and they'll be normal because they have X big C Y. 25% will be female and they'll be normal, and 25% will be female and they'll be carriers with the big C small c.
Codominance is when both alleles are expressed in an individual. The example here we use is red snapdragons.
So we've already looked at codominance, but they're very interested in how it works from a blood group point of view. So there are 4 different blood groups. They are A, B, AB, and O, so you need to learn those. You also need to learn the genotypes which represent each blood group, and from a codominance point of view, remember that we have got to write them in this very specific way, which is we choose the base letter and then we have a superscript showing the allele for the particular blood group, and that will be more clear once I start drawing them out. So for A, the potential genotypes are IA, IA, or IA, IO. For B, you're looking at IB, 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 IO. For AB, you're looking at IA and IB. And for O, you need IO, IO. Just make sure you're clear that that's an OR. And now let's have a look at an example so you can actually see it in practice. So let's look at an example to make it nice and straightforward. So we're drawing a genetic diagram to explain the inheritance of blood group in the Smith family. Mr. Smith has the genotype IA-IB and Mrs. Smith has the genotype IO-IO. So using my method from before, we're going to lay it out as a table. So Mr. Smith, Mrs. Smith. Start with our phenotypes. So what blood groups do they have? Well, according to my previous slide, IA-IB will obviously be blood group AB for Mr. Smith. Mrs. Smith has IO-IO, which means her blood group is O. So what are their genotypes? Well, happily that's been given to us in the questions. So that's just IA-IB for Mr. Smith, IO-IO for Mrs. Smith. So the gametes, we just need to separate those alleles and draw circles around them to show that this is the potential alleles for his sperm, and her eggs will all be IO. Now let's do the Punnett square. And just cross them. And then from the previous slide we know what each of the child's phenotype will be. So IO-IA, that's 50% of the offspring, and they'll have blood group A. And IO-IB is the other 50% of potential offspring, and they'll have blood group B. So now we need to look at pedigree diagrams, and the best way to do this is by showing you an example. Always use this approach, and do notice these are supposed to be really difficult, so don't worry too much if you're finding it too much. Question 3. Familial hypercholesteremia FH is an inherited condition caused by a dominant allele. That is key. People with the condition have high levels of cholesterol in their blood, increasing the risk of dying from blocked arteries. The diagram shows the pattern of inheritance in several generations of a family with familial hypercholesteremia. So do notice with the pedigree diagram that the squares are always the men in the family. You'll know this from the key. The circles always represent females. And in this case from the key, we can see that the grey shaded boxes are sufferers of FH, whilst the white boxes or circles are non-sufferers. So person A is heterozygous for FH. Use this information to complete the table. So let's start by labelling the genotype of person A, and we are going to use the letter D. I mean, it doesn't matter what letter you use, but I'm going to use the letter D because you can easily see the difference between a capital D and a small d. So labelling their genotype, this is what they look like, so big D, small d. So what is the question actually asking? How many people have the genotype which is homozygous recessive? So homozygous recessive, homozygous meaning the same case, recessive meaning lowercase, which is why we're looking for small d here. Homozygous dominant, homozygous meaning the same, dominant meaning that they're both capital. So that's what we're looking for. Now we're going to work out what the pedigree diagram tells us. First of all, I'm going to look at all the people without FH, so all the people that are either white circles or white squares. Because they don't have the disease, I know therefore that they are small d, so I can just label all of their genotypes straight away. And now we need to count them to work out the number of people with the genotype homozygous recessive. And once I've done that, I can see that it is 11. Now we're getting slightly more difficult by looking for the big D, so homozygous dominant. So we need to infer things from the pedigree analysis. First of all, look at woman C. So she got her genotype from parents A and B. Now B is homozygous recessive, which means they must have passed on a small d. She has the disease, which means she must have a big D. So this is her genotype. E has the same issue, but they're a man, so they're going to be big D, small d. Person G inherited a small lowercase allele from D. They have the disease, which is why they're capitalized. And the same goes for person J. And then looking at NOP, well, they inherited a small d from their mother. They have the disease, which is why they have a big D. So actually, looking for people with big D, the answer here is 0. Person G and H have 3 children, all of whom have FH. What is the probability of G and H having 3 children who all have FH? This is a crazy amount of work for 1 mark, because the only way I can see of doing this is to draw a Punnett square. So we're going to do a Punnett square for G and H using my layout I already described. So we're looking at the phenotype, genotype, and gametes. From the key, we can see that person G has FH. H is therefore healthy from the key. We've already labelled their genotypes, so we can just copy that directly across and then separate these out to see the gametes. Now just simply do a cross, and it's these 2 here that will have FH. Now what is 50% as a probability? Well, it's 0.5, and the question asks the probability of all 3 children having FH. So remember, when we're talking about probability, we have to multiply together our probabilities. Pop that into your calculator and you'll get a value which is 0.125.
Very short topic now on evolution and natural selection. So first of all, your definition for evolution. It states that many organisms which are alive today and many more which are now extinct first evolved from very simple life forms that first evolved over 3.2 billion years ago. So that's basically saying that evolution states that we all evolved from small life forms like bacteria, which became multicellular, which became more and more complicated. They became reptiles, they became birds, and then they became mammals, and then we came about. So that's really what evolution is stating. Natural selection links very nicely with this. Remember, this is Charles Darwin's theory. Now, he stated, and I do just want you to learn this as a 5-mark answer off by heart, he stated that there is variation within a species due to mutation, which is what I've just discussed. So within a species, there is variety. This means that some individuals within the species are more likely to survive because they are better adapted. Because they're surviving, they're likely to reproduce, so produce offspring, and those offspring will inherit those favourable genes.
So before you know it, you have many generations that go past and they've all inherited this favourable gene, making them more likely to survive. And I'm now going to bring up that perfect answer for you. Natural selection can be seen pretty much everywhere on Earth, including bacteria. So we're just going to describe how bacteria may become antibiotic resistant, and it does link to natural selection. So what happens is you have a colony of bacteria, you give them an antibiotic, and due to mutations, some of those bacteria are stronger, they are resistant, that means they are not killed by the antibiotic. So what happens is all the other bacteria are killed, leaving behind these very strong antibiotic resistant bacteria. They soon replicate, and before you know it, you've got a colony of bacteria which is no longer treatable using antibiotics. And that's why everyone's so scared about antibiotic resistance and what it means for our future medicine. So how do stone tools provide evidence for human evolution? Well, let's first of all look at the evidence of human-like species using stone tools, and that actually occurred 3.3 million years ago, so a seriously long time ago. And they noticed that the more recent tools showed much greater sophistication. So basically, tools became more sophisticated over time. And how do we know how old these things are? After all, we went around 3.3 million years ago. This is where physics comes in, and particularly radioactivity, because remember, we can do radioactive dating. Looking at the pentadactyl limb now, so pentadactyl, so pent meaning like pentagon, so 5. Dactyl is to do with how many fingers you have, so that's 5-fingered limb. And really, we like to use the anatomy of the pentadactyl limb to provide evidence for evolution, because actually you find the same limb anatomy, so the same basic limb structure, between all kinds of animals, including turtles, dolphins, bats and humans. And I know you're thinking, but hang on, we look completely different from a dolphin. However, the point is, we think that we all evolved from the same common ancestor, and then the various branches came off, and the pentadactyl limb ended up evolving into a completely different structure because it needed to serve a different purpose. So obviously for dolphins, it needed to become a fin to enable them to swim, whereas for bats, that 5-fingered limb needed to become wings to help them to fly. And then obviously as humans, we needed them to become hands to help us pick up stuff. And that really sets us apart from other creatures. So the suggestion is that we came from a common ancestor. Now we know that millions and millions of different creatures exist. So how do you know if one creature you found is a new species or if it already exists but you just haven't clapped eyes on it yet? So we use a classification system in order to help us name these things. And one of the original classification system was Linnaeus's. I don't know if I'm pronouncing his name right. And he basically said that there were 2 kingdoms, the animal and plant kingdom, and that these were divided into smaller and smaller groups based on their physical characteristics. However, the issue is with his particular classification system is based on putting animals into groups based on their physical characteristics. You'd end up putting bats and ladybirds or flies in the same group. And the difficulty here is if you think about a bat wing versus a bee's wing or a fly's wing, it is so unbelievably different. And that's because the evolution of their wing came about by completely different processes, so it wouldn't be right to place them in the same group. Plus, he only includes animal and plants, and we know that lots of other type of organisms exist, such as fungi, protoctists, prokaryotes, etc. So really, his system wasn't big enough. So the traditional classification systems, so not ones based on the binomial method we use now, was based on evolutionary similarities, and it grouped organisms based on their morphology and anatomy, so effectively the bones which made them up. A more accurate way of classifying these organisms is to look at greater detail, so actually look at their structures. That means looking at the sequence of bases in their DNA and how these sequence of bases determine which amino acids form which proteins. So that's a more accurate way of determining how we should group organisms. More similar sequences of DNA, so more similar amino acids and proteins, are more likely to be closely related. If we're going to use the correct nomenclature when we're talking about naming things, we can talk about the 5 kingdoms, and that consists of plants, animals, protoctists, bacteria and fungi. Now, selective breeding. So remember, this is when humans use animals or plants with desired characteristics, they force them to breed, and then they repeat this process over many generations. So before you know it, you have animals with desired characteristics. So if we're looking at animals, let's, for example, look at the dairy industry. So dairy cows, clearly a good animal here will produce a lot of milk. So humans, so make sure you point out that it's humans, they select a dairy cow that produces a high yield of milk. They mate her with a bull. It's quite hard to determine the bull because obviously they don't produce milk, but they'll mate her with a bull. And then her calves are likely to produce more milk, the female calves, because of their high yield mother. Then you take those calves and you keep repeating the process until you have lots of calves and lots of cows that produce lots of milk. And you can do the same with plants. So you can selectively breed plants to be a particular colour. So you pick flowers that are a particular colour, you force them to cross-pollinate, and then before you know, you've got a load of plants with your desired characteristics, such as petal colour. Genetic engineering now. This is quite a complicated topic. It is chock-a-block full of key scientific words, but if you learn them off by heart, you should be fine. So remember, we genetically engineer things like insulin. So insulin is a hormone produced by our pancreas, and it's responsible for lowering our blood sugar levels after we've eaten. And for type 1 diabetics, they find that they don't produce insulin, so they really struggle to maintain their blood sugar levels, which is where genetic engineering comes in, because in the olden days they used to obtain pig insulin. So they used to chop into pigs, remove the insulin, and not only do you have major ethical issues with this, but obviously the insulin wasn't particularly fit for purpose because it came from pigs. So it was important that we found a way of producing insulin from humans, and so that's where genetic engineering came in. And when we're talking about genetic engineering, we're talking about using bacterial cells, because bacterial cells contain small rings of genetic information called plasmids, which we can manipulate so that you can insert the insulin gene and force the bacteria to produce lots of insulin. So let's go into great detail how that is done. So we obtain the bacterial cell and we cut open the plasmid using a restriction enzyme, which acts as a pair of biological scissors. Then we use a restriction enzyme to cut the insulin gene away from the rest of the cell, and we insert that insulin gene into that bacterial plasmid using a ligase enzyme, and we stick it together, and that's why we say it has sticky ends. Once we've done that, we're ready to put the bacterial cell into a fermenter, and it's been done many times. And then I've already mentioned the fermenter. You need to provide it with the optimum conditions, so the right temperature, the right pH, the optimum amount of oxygen and nutrients, etc. And before you know it, your bacteria has made huge amounts of insulin. Don't forget a few keywords here concerning genetic engineering. Once that plasmid has a different gene inserted into it, we call it a recombinant plasmid, which means it's been recombined, so it's been changed. And don't forget also that the bacterial cell, the plasmid, is acting as a vector, which means it transports biological material from one place to another. We can also genetically modify plants so that they can have desired characteristics. This could include being frost resistant, so that stops them dying when frost hits in winter. It could be to extend their shelf life, to stop them going off so that they have a longer shelf life and are more fit for human consumption after many days. You might actually want to make plants resistant to weed killers. This sounds really strange because why would you want to make a plant resistant to weed killer? But think about it. A farmer applies the weed killer. By the way, the best name for weed killer is herbicide. Side meaning kill, herb meaning to do with plants. So the farmer applies the herbicide. He or she wants to kill the weed. However, some of it will inevitably fall on the plant that they're trying to grow. And obviously you don't want that to happen because it will actually kill the plant you're trying to grow. So if you can make that crop plant resistant to herbicide, then that's great because you apply that herbicide or weed killer, it kills the weeds and your actual plant that you're after stays alive and continues to grow. You can also genetically modify plants so that they can actually have health benefits. One of the most famous examples of this is golden rice. So when poorer countries grow lots of rice, unfortunately rice doesn't have a huge amount of nutritional value. So what you can do is genetically modify it so that it contains vitamin A, and therefore when people eat that rice they get a huge amount of vitamin A in their diet, and that stops them getting night blindness. This is something you should remember from the balanced diet topic of the specification. So golden rice is an excellent example where genetic modification has been used really well. And then the most strange example of genetic modification is when we talk about tobacco plants, and these have been modified so they actually produce hepatitis antigens, therefore have a potential vaccine against hepatitis. So this is like crazy science, but just remember that tobacco plants may be modified to produce hepatitis vaccines. So we've already touched on this, we've given lots of examples of genetic modification in plants. Just to reiterate the advantages and to throw in a few more, you can have increased salt tolerance, So how does using fertilisers increase crop yield? Well, the addition of fertilisers to the soil replaces leached or lost nitrates and mineral ions from the soil, because remember fertilisers are very rich in nitrogen, nitrates, and those nitrates are used by plants to build proteins. What is a pesticide? Remember, it is a chemical which kills pests, so anything which feeds off plants will be counted as a pest. Killing pests obviously reduces their damage to the crop, and it also helps to increase crop yield. Looking at how we control pests further, remember we can add chemicals, so pesticides, or we can use biological control, which is about using other animals which kill and eat the pests, and we need to look at their various advantages and disadvantages. So let's first of all look at the advantages of using pesticides. Now these are easy to use, so they're easy to apply, they're effective, which means they do a pretty good job of killing the pests, and they're readily available. Issues though, there's lots of issues with using pesticides. Firstly, that they can be very expensive. They're persistent, which means it takes a while for them to decompose, so once you apply them to your soil, you've got to be aware that they may hang around for many, many years. And the problem here is that they can often kill animals which aren't even pests, which is really, really bad, because these are innocent animals getting killed by the pesticides because the pesticide does not discriminate correctly. So what happens here is it kills other animals. Some of these animals get eaten by larger animals, so we're talking about food chains here. And this is called bioaccumulation, where the pesticides become stored in these animals, and then as this pesticide works itself up the food chain, we call this by magnification. And the famous case study of this is DDT, which was used to eradicate malaria and typhoid in the Second World War, and there are still areas of the world where DDT is killing huge amounts of flora and fauna, and that just means animals and plants. Another disadvantage is that you have to keep reapplying this pesticide. Looking at biological control now, so let's name a few examples. So like I said before, this is using animals to kill pests. The most famous one probably is using ladybirds, because ladybirds are predators to aphids, so they come along and munch on the aphids, which would otherwise be destroying things like cabbages. So what are the advantages of using ladybirds using biological control? Well, they tend to be quite specific and kill the pests that you're after. Secondly, they're self-sustaining. They tend to reproduce, which is great because you don't have to keep reapplying ladybirds. They tend to grow into new populations which will continue to eat the aphids, and clearly they'll be non-toxic, especially when compared with things like DDT. However, there are disadvantages. They have been known to not just eat the pests that you're after, they can go around eating other things, so that can be both an advantage and a disadvantage.
They never fully eradicate the pest, so there will still be some aphids which survive the purge because the ladybirds don't go and eat all of them. When you add extra animals to an environment, to an ecosystem, they can have undesired effects. You don't really know what they're going to do. They can have major effects on food chains, they really disrupt them. So you do have to be careful before you decide to apply these animals to your ecosystem. And lastly, compared with using pesticides, it's pretty damn slow waiting for ladybirds to go and eat all the aphids, whereas with pesticides you tend to find that everything gets wiped out immediately.
Now we need to look at the health, disease and development of medicines topic, and starting with the WHO's definition of health. And WHO, or WHO, is the World Health Organization. So people tend to think that they're the go-to in terms of knowing about these things. So what is their definition of health? Well, it's a state of complete physical, mental and social well-being, and not merely the absence of disease or infirmity. So it's not about being like, right, I haven't got a cold, I haven't got this, I don't have this particular genetic disorder. It's talking about both your mental and your physiological health. Let's look at what physical well-being really means, and it means that no disease, so an absence of disease. It means good diet, regular exercise, and a low intake of harmful substances. So that could be dangerous drugs, for example. Looking at mental well-being, really we're looking for happiness and contentment in being who you are. And if you have both of these things, you have complete health according to WHO. What is social well-being? That's to do with your relationships with other people. So we're going to write relationship with other people is good, and that you're comfortable in your surroundings. So is there a relationship between health and income? Well, lots of people tend to think there is. So if we were to draw a really basic graph showing increased income alongside increased health, you would definitely see a positive correlation. Obviously, this is a very general statement and it's not necessarily true for everyone, but as income increases, health increases. And that's for obvious reasons, in that if you have a higher income, you have better access to medicines and better diets. And places where people tend to have higher income occur in countries which are less prone to natural disaster, because natural disaster like earthquakes obviously kill large numbers of people. Notice that the relationship between health and income isn't causative. So it's not necessarily true that you have a higher income, meaning that you have greater health. Looking more closely at disease now. So what is a disease? Well, it's a disorder of the body which produces specific symptoms and crucially, it's not a result of physical injury. So if you were to damage your leg having fallen down the stairs, that is not an example of a disease. Now, there are different types of disease you need to know about. First type is communicable diseases. And as the name suggests, these can be transferred between people. And these tend to be caused by pathogens. And remember, a pathogen, if they ask you, is a microorganism which causes disease, such as a bacteria or virus. Now, a non-communicable disease is one which cannot be passed readily from person to person, and it's actually caused by your genetics or lifestyle. So if you inherit cystic fibrosis, that is a non-communicable disease. If you get lung cancer due to smoking, again, a non-communicable disease. You can't pass on lung cancer by accidentally brushing against someone, for example. Let's touch more on this pathogen definition. We've already said what it is, but there are other words associated with it that you need to be aware of, and one of those is a vector, which is actually a maths term as well, but in biology it means an organism which carries pathogens. So it doesn't actually cause the disease itself, it's just a means of transporting a pathogen from one person to another, and a good example here is the mosquito. So although these are super annoying when they bite you and suck your blood, they're not actually responsible for giving you malaria. In fact, it's the fact that they carry a protoctis called Plasmodium, and it's obviously an example of a pathogen, and that's actually what causes malaria. Viruses are very different to bacteria and other types of cell, mainly because they're non-living. They simply consist of DNA or RNA surrounded by a protein coat. And they need to reproduce inside another living organism. Many diseases interact, and if you are suffering from one disease, it can mean that you increase your likelihood of getting another. And why is that? Well, that's because your immune system becomes weakened, and therefore other pathogens are more able to cause disease. When you're suffering from a disease, it does tend to mean that your various barriers may become damaged, meaning that it's more likely that pathogens will be able to enter. For example, if you have measles, it's a very, very bad skin rash. In fact, it can be fatal for some people. Now, you're likely to scratch because it's itchy, and obviously if you scratch your skin, you break that barrier, meaning that other pathogens can enter your bloodstream. And lastly, if the disease affects various important organ systems within your body, it does mean that other diseases are able to occur. For example, if you got tuberculosis, well, that would damage your respiratory system, so your lungs, and therefore you'd be more likely to get something like bronchitis. Now we need to look at various types of pathogen and the diseases they cause. So let's start with a bacteria called Vibrio chlori. And as the name, hopefully you can see that that would cause cholera. And unfortunately, people that suffer from cholera get chronic diarrhea, which can actually kill them. It's a really terrifying disease, and it's a waterborne disease, so the bacteria live in infected water supplies. Looking at another example now, I've already mentioned this one, tuberculosis, so I'm going to call the disease here TB. It's caused by the Mycobacterium tuberculi bacterium. Some nasty symptoms here. You often end up coughing up blood, and that's due to lung damage. These bacteria make small holes in your lungs. The Ebola virus now got a lot of press a few years ago. It was killing lots of people in Africa. This causes hemorrhaging, so bleeding from all kinds of orifices, and we call this hemorrhagic fever. So hopefully you recognise heme, that means to do with blood. HIV now, human immunodeficiency virus, which progresses to form the disease AIDS. This destroys the immune system, it infects your white blood cells. And lastly, a protoctist by the name of Plasmodium, which I already mentioned. causes the disease malaria, and it causes widespread damage to your blood and liver, and even your brain sometimes, depending on the type you get. Let's label these various pathogens and talk about how they're actually spread. So Vibrio chlori, I already said, its mode of transmission is infected water. So if people drink water containing Vibrio chloride, they will get cholera. Mycobacterium tuberculosis, TB, maybe you know this from history, people got it from accidentally coughing on each other, which means that its mode of transmission is that it's airborne. Ebola is spread in infected bodily fluids, so blood carries it, and we know that you get excess bleeding with Ebola, so if you accidentally get some of that infected blood on you, that's how you could potentially get Ebola. HIV is a sexually transmitted disease, so if you have unprotected sex, you're more likely to get HIV. And we've already said that Plasmodium is spread via the vector mosquitoes. Some more key definitions now. So what is a host? Well, it's an organism carrying the pathogen and the disease. So if it infects us, we'd be the host. If it's a plant that houses tobacco mosaic virus, then the host here is the plant. So we've talked a lot about various types of disease and how they're transmitted, but how can we actually prevent them? So number one, just generally good hygiene, washing regularly in order to remove those pathogens from our body. To prevent airborne diseases, you want to avoid coughing and sneezing on people, which is why I find it particularly gross if people accidentally cough or sneeze on you when you're on the tube. If anyone's been to London in rush hour, you will know what I'm talking about. To prevent diseases spread by dirty water, you want to treat that water, so disinfect the water to kill pathogens. Diseases such as HIV, which is spread through sexual contact, you want to wear condoms in order to prevent the spread of HIV. And lastly, if you're trying to prevent a disease which is spread by a vector such as mosquito, clearly you want to kill the vector, or you want to drain its water supplies where the vector reproduces, or you want to cover your beds with nets to stop the mosquitoes biting you. We've already mentioned how viruses are so very different from a regular animal, plant or bacterial cell, but how do they actually infect an organism? Well, firstly, they have to inject either their DNA or RNA into the host cell. Next up, they need to hijack the living processes of the cell so that the cell starts to do the virus's own bidding. As part of this, the cell copies the viral DNA, and then effectively the cell bursts, releasing thousands of copies of the virus, which can obviously go on and infect other cells, and before you know it, the virus has spread everywhere and has caused massive damage to pre-existing cells. A key definition now, which is lysis. Lysis is simply the total breakdown of a cell. It ceases to live. Linked with what we were saying about how viruses replicate, and the meaning of the word lysis is the lytic cycle. And this relates to viruses. And it's actually the process by which a virus replicates itself and destroys the host cell. A second important definition is a phage, and this is a virus which particularly infects bacterial cells. So pathogens go out of their way to cause disease, damage us as humans and other animal hosts and plant hosts. So how can plants, for example, defend themselves against attack? And there are two main ways. The first one is physical barriers, and the second is chemical barriers. So let's list some physical barriers. Firstly, the waxy cuticle forms a tough outer casing on the leaves and the stems. Oak trees have a particularly thick bark. And generally the cell wall of the plant, remember it's made out of a tough material called cellulose, which acts as a protective measure. Now we need to look at chemical barriers, and we're really looking at poisons. Many plants contain some pretty toxic poisons which can kill insects if they try and eat them. And that's why it was really careful back when we were hunters and foragers. We had to make sure that we didn't poison ourselves by eating deadly plants such as the foxglove. Foxglove is very famously poisonous, so don't touch those if you see them in woodland. But as well as being poisonous, a lot of plants contain some very useful chemicals which we can actually use to make drugs, used to treat disease, and pretty famous ones include aspirin, which was actually originally produced from the willow tree, and that's used to relieve pain and help to thin the blood if you're prone to having a stroke. Also helps reduce inflammation. Slightly more niche is artemisinin, which is used to kill the plasmodium, which we know causes malaria. So how can disease actually be detected in plants? First of all, just by looking at them, so we observe the symptoms. Such as if a plant's suffering from tobacco mosaic virus, you get a very distinct discoloration of the leaves. You can also use home testing kits, or you could send off your sample to a laboratory for analysis. Linked with this is a fairly niche topic, which is distribution analysis. And this is an analysis of where the damaged or diseased plants are located, and you often find that similar symptoms can be caused by a variety of factors, such as diseases spread by wind have different distribution than diseases caused by pests or lack of nutrients.
Next topic, white blood cells and the immune system. So let's first of all discuss how we prevent pathogen entry in the first place. So remember, our skin acts as a barrier. Our hydrochloric acid in our stomach helps to destroy pathogens. Our tears prevent pathogens entering our eyes, and also your eyelashes. But what happens once those pathogens actually enter our body, enter our bloodstream? Clearly we can't stay ill forever and ever and ever. So there are mechanisms in place which actually act to remove those pathogens. The two mechanisms you need to know about are white blood cells, and they are the phagocytes and the lymphocytes. So starting with the phagocytes, remember that they engulf or ingest pathogens by enclosing them inside a vacuole, and then digestive enzymes are secreted which destroy the pathogen. The second type of white blood cell is the lymphocyte. The lymphocyte is far more complicated, and it works by recognizing the antigen on the pathogen. It secretes lots of antibodies which destroy that specific pathogen, and in this way the pathogen is destroyed. Now, it has various modes of action which help to increase the pathogen destruction. First of all, it labels the pathogen, making it easier for the phagocyte to recognize it and therefore engulf it. It neutralizes any toxins produced by the pathogen, and it also causes the bacterial cell to burst open on occasion. Lastly, it makes the pathogens stick together. With that answer relating to the lymphocytes, notice that I use lots of keywords, antigens, antibodies, for example. Try and include as many keywords as possible. Just shove them in your answer, because if you look at mark schemes, they'll be underlined as being worth a mark each, so it's worth writing them in anyway. Don't just keep repeating yourself. You need to insert lots and lots of keywords here.
Lastly, vaccinations, vaccines. So obviously when you're going on holiday somewhere tropical, you might need to go to the doctor to get some vaccines. Now what these are is they're injections containing either a dead, weakened or attenuated form of the pathogen. That means it contains the pathogen's antigens. Now when those antigens enter your body, clearly your lymphocytes are going to be set off and they're going to produce antibodies which actually respond to those pathogens, and some of those lymphocytes turn into what's called memory cells. Those memory cells remain in your body, and therefore if you become infected at a later date with a much larger amount of that pathogen, there are memory cells already in place which can secrete antibodies very quickly, much faster, much sooner and in a much larger quantity, and what that means is that pathogen can be destroyed before it can take a hold of your body. So the whole point of vaccination is to inject a harmless version of that pathogen so that if you accidentally become infected at a later date, it can be destroyed before it can take hold. So let's look more closely at some vaccinations which are made. So dead pathogens are used to immunise against whooping cough. We used a weakened form of the pathogen to treat measles and also tuberculosis. And then lastly, we just inject the antigens themselves when we're treating influenza. Herd immunity is just a term describing when a large number of people get vaccinated against something, because something like measles virus will need a large number of people to infect. So it will infect a person who will then go and infect another person that will go and infect another person. And that's how the measles virus is spread. Now obviously, if you go and vaccinate loads of people against the measles virus, there are going to not be enough people for the measles virus to spread properly between. And that's how you actually get herd immunity against something like measles. It basically means when a large number of people are vaccinated against a specific disease. If you want to kill a pathogen like a bacteria, you're going to have to use an antibiotic, and antibiotics only work on bacteria. They do not work on viruses. So I've already described one problem with antibiotics, which is that they're only effective against bacteria. The second problem is that increasingly we're seeing antibiotic resistance. And that's what happens when we don't complete the course of antibiotics that we're taking, if we overprescribe them, because you find that you'll get some strains of bacteria which are actually resistant to the antibiotic, and then before you know it, all the new bacteria that are being made are resistant. So back in the day when a certain antibiotic would work, you're finding later that it doesn't work, and obviously that's going to cause massive issues if we don't have antibiotics that work.
Now in a laboratory it's really important that we use aseptic techniques, and that's basically to prevent contamination by microorganisms, and there's lots of different things we can do, and that includes sterilising a variety of equipment. That could be using an autoclave, which is a special machine manufactured in order to sterilise. It could be just using a hot flame. And if you've done any work at school, maybe using Petri dishes, remember you're supposed to cover the Petri dish straight away to prevent airborne contamination, and you need a sterile growth medium. So if you're providing your whatever it is you're trying to grow, like that bacteria, you need to provide them with a sterile agar jelly. So let's make notes on that now. So why do we use aseptic technique? Well, fundamentally it's to prevent contamination by microorganisms. And how can we do that? By a variety of sterilisation techniques. So we can sterilise our equipment using an autoclave. When we're using Petri dishes, we use sterile agar jelly. And lastly, again using Petri dishes, we cover our Petri dishes when not in use to prevent airborne contamination.
Next up, we need to look at Petri dishes. The Petri dish is a little plastic dish which we use to grow bacteria on, because we're interested in that bacteria, how it grows and how we can actually prevent its growth using antibiotics. But in terms of preparing a Petri dish for inoculation, first of all you need to get an inoculating loop, which is just a metal loop that you effectively swab the bacteria onto, but you first of all need to disinfect that loop to make sure there's no bacteria pre-existing on it, and you need to dip it into a blue flame of a Bunsen burner, again to ensure that it's totally sterile. At this point you're going to swab it on the bacteria sample that you're interested in, and you're going to wipe it across the agar jelly that's inside your Petri dish. Now the agar jelly is a nutrient medium, which means that it's full of food that the bacteria can feed on. At this point, you're going to place the lid on the Petri dish, and you're going to just make sure that some air can get into that Petri dish to allow the bacteria to aerobically respire. And then you want to seal it with tape just to make sure that nothing spills, and that is how you're going to set up your inoculation and your Petri dish.
A small bit where we dip into maths now. So remember, if you have a Petri dish, so we're looking down on it, and then we put various antibiotic discs down, and I'm going to call them A, B, C and D. Now the bacteria start to grow, and then if they reach a very effective antibiotic, let's pretend B is extremely effective, then they can't grow around that disc. Say A is a not very good antibiotic, so the bacteria can grow pretty close to that disc. D, we're going to say, is an excellent antibiotic, so it's going to have a huge clearance zone. And then again with C, we're going to say that it's not a very good antibiotic for this particular bacterium. It hasn't managed to kill much of the bacteria. So as we can see, very little clearance, and the green is obviously the bacterial growth. So how can we actually work out that clearance zone? Well, it's a roughly circular shape, so we're going to calculate the areas. And for maths, how do we calculate area? By doing pi r squared, and r is the radius of the circle. So if we take the clearance circle for D, this would be r, and then we need to square it and times it by pi to work out the area. And as I said, this is used to analyse the effectiveness of an antibiotic on bacterial cultures. And the larger the area, the more effective the antibiotic.
Let's talk about how we actually go about developing drugs then to treat ourselves. So in terms of what we're after from a drug, obviously you want it to be effective, because what's the point of taking a drug if it doesn't actually work? You want it to be safe, so you don't want to have it causing any adverse effects on your health, because that wouldn't be great. You want it to be stable, and what that means is that it needs to be easily stored. You don't want a drug that can only be taken within like a week and then goes off in the cupboard and starts causing like problems. You want it to be nice and stable. And lastly, the least obvious one, it needs to be successfully taken into your body, so easy to deliver, whether that's drinking it or having it injected, and it needs to be able to be removed from your body too. I don't know if you know this, but lots of drugs come from plants, and lots of these plants are found in the rainforest, and obviously you can't get some chemical from a plant and then just give it straight to someone, because you don't know the effects it will have and it may kill them. So there are lots of stages involved in the development of a drug. The first stage is preclinical trials, and that's when they test a chemical or a drug out on tissues or cells or other animals, and that's where animal testing comes in, and that's why lots of people have lots of problems with animal testing, and I understand that. Once they've tested it out on the animals, on other cells, they will then start testing them out on human volunteers, and we call that clinical trials, and they'll start by giving human volunteers very, very small amounts, so that if there are going to be bad effects, hopefully they're not going to be too dangerous, and then they'll increase that dose, the amount that they give the person, bit by bit, until they're happy that it's the right amount. When you're talking about actually doing trialing, there's something called a double-blind trial, and that's when neither the doctor nor the patient know whether they're being given the drug or not. And that's essential, because a lot of times if a patient is given a drug and told that it's really going to work, they actually kind of get themselves better through their own mental state of mind. They'll be like, wow, I've been told this drug will work, so I'm going to get better because of it. And that's obviously giving you a false positive result, because it's not necessarily the drug doing that. So a double-blind trial is when neither the patient or the doctor actually giving the drug know whether the patient's had the drug. Because the other thing they could be given is a placebo, and a placebo is just a tablet that's like made out of sugar that has no drugs in it. So some patients will be given a placebo, some will be given the drug, and then you'll look at the results to try and work out how effective that drug is. Now we're getting onto the tricky topic of monoclonal antibodies. So what we're talking about here is a new development, it's a new technology, and we need to first of all understand what the word hybridoma means. And all that means is it's a new cell type formed by combining mouse cells, human cells, and cancer cells in order to form a new type of cell. So I've already told you that lymphocytes will produce antibodies which will go ahead and destroy pathogens, but the problem with lymphocytes is they don't actually
divide and grow. So in an ideal world, you'd get a load of someone's lymphocytes, cause them to divide to make loads of antibodies, and then go around treating people using those antibodies. But I've just said that lymphocytes don't divide. So what we need ideally is a type of cell which divides rapidly, and in this case we know that cancer cells divide very rapidly because they go ahead, they form tumours. That's a really terrible thing about cancer cells, is these tumours that grow and obstruct parts of our organs. So if they're in our brain or our heart, that's where all the issues arise from with cancer. So if you can think you could get a lymphocyte that's producing all those awesome antibodies that will go and kill stuff, but then you've got the cancer cell which has the ability to copy itself and divide lots. Imagine if you combined them, then you'd have the perfect situation where you're going to generate lots of antibodies that will go ahead and kill stuff, but won't be cancerous and therefore won't cause any problems. And that's really what this monoclonal antibody stuff is all about. Now, what does the term monoclonal antibody mean? Well, it's just a protein which is made to target specific cells. So the crucial thing here is they're very specific. Now, they found that mice lymphocytes, so that little mouse, they found that lymphocytes inside the mice are amazing at producing these monoclonal antibodies. So they combined it with the cancer cell in order to form this hybridoma, which I've already mentioned, and then that could go ahead producing all the antibodies needed to destroy dangerous cells. I mean, the obvious problem with this is that they come from mice, and obviously our bodies are going to recognise mice cells as being foreign, and then our immune system will try and attack those mice cells. So what they then did was they combined it with human cells in order to prevent the rejection issue, and that's why the hybridoma is a combination of mouse, human and cancer cells. So how can monoclonal antibodies be used? Well, firstly, pregnancy testing, because there's a hormone called HCG which pregnant women make. Now what happens is the monoclonal antibody can actually bind to that hormone, and then it gets expressed in urine, which can then be detected. So it's actually impossible to get a false positive result. You will not get a false positive result. You will definitely be pregnant if you get a positive result. So I think that's quite an essential life point right there. The second thing that you can use monoclonal antibodies is for detecting disease, because it can actually bind to disease cells, and that's used to detect prostate cancer in men, for example. And lastly, you can use monoclonal antibodies to detect levels of hormones. So an example of this is, you know how people donate blood, because blood donations are really important, but obviously you don't want someone with the disease AIDS or HIV donating blood, because that could go and infect the poor people that the blood's being given to. So monoclonal antibodies can actually be used to screen that blood and see if there's any HIV in there. Really, really good stuff. Now, how can monoclonal antibodies be used to treat cancer? Because really that's what we're after. That's the really amazing role that they perform. So first of all, they can help carry toxic drugs to the cancer cells, and therefore those toxic drugs can go to work on the cancer cells without affecting our normal cells. Secondly, the monoclonal antibodies can help trigger our immune system to actually go and attack those cancer cells, whereas before it might not have been doing that. And thirdly, those monoclonal antibodies can block the receptors on cancer cells, and those receptors are needed to help that cancer cell divide to form a tumour. So if it blocks the receptors, they can't divide anymore, which is obviously essential to prevent tumour growth. Now we need to look at the advantages of monoclonal antibodies. The first thing is that they have no effect on healthy cells, and that's crucial because a lot of cancer treatments such as chemotherapy and radiotherapy do go and damage a lot of healthy human cells, which is obviously not what we're wanting. The second thing is that the monoclonal antibodies can be used to treat a wide range of conditions. However, sadly, there are disadvantages. That is, some side effects can arise from using monoclonal antibodies. And actually the technology involved, it's been tricky to develop, much trickier than they first imagined when they first started developing this.
So let's look at some examples of some key non-communicable diseases. We've already touched on a few of them, but I want to create quite a large list here. And remember, these are diseases which are due to our lifestyle choices, due to genetics. They cannot be passed on from one individual to another. They can't be caught, for example, by airborne transmission or waterborne transmission. So we've already mentioned genetics, so faulty genes. And there are lots of examples of genetic diseases such as cystic fibrosis, sickle cell anemia, Huntington's disease. Another type of non-communicable disease is due to malnutrition or a deficiency disease. So this is when you don't have a balanced diet and your diet is deficient in particular minerals or food groups. So if you don't have enough protein, you get a disease called kwashiorkor. Marasmus is also due to a lack of protein. Scurvy comes about due to a lack of vitamin C. And anemia is a result of a lack of iron. And that makes you feel pretty exhausted. Looking at lifestyle choices now. So what sort of unhealthy habits do people develop and what kind of diseases does that give them? So if you smoke, you're more likely to get lung cancer. If you drink too much alcohol, then you give yourself liver disease and that could lead to liver cirrhosis. Obesity, so overeating, can lead to diabetes type 2. And touching more on liver disease, particularly in the UK, it is very common. It's actually the 5th highest cause of death in the UK. And a large part of that is due to alcohol consumption increasing. So we looked at obesity, but how do you decide if someone is obese, whether they're morbidly obese or if they're a healthy weight? Well, we have different ways in which we can determine this, and one of this is calculating the BMI, and the BMI stands for the body mass index. And you calculate that by finding the person's mass in kilograms and dividing it by their height in meters, and you need to square that. And in terms of the values, if you have a BMI of over 30, then you're classified as medically obese. However, it's not a perfect way of measuring whether someone's obese or not, because people, for example, that work out a lot and build up lots of muscle, they're going to be correspondingly pretty heavy and therefore register as obese when clearly they're just muscled.
Looking at coronary heart disease now, the coronary arteries. Coronary means relating to the heart. So the heart has its own special network of vessels which supply the heart with its own supply of oxygen. It can't actually obtain its oxygen needs from the blood flowing through it. It has to have its own special set of vessels. We call these the coronary arteries, and they're famous because this is how people get heart attacks. They get blocked, they get obstructed, and it does mean that oxygen can't reach the heart muscle, so part of it dies, which is what a heart attack is. So first of all, what factors increase your chance of getting coronary heart disease? So that could be a sedentary lifestyle, so lack of exercise. It could be your diet, eating diets high in fat and sugar. It could be inheritance, so genes. Some people are just more susceptible than others because of genes that they've received from their parents. It could be diabetes. Diabetes and coronary heart disease are very closely linked. Stress as well. People shouldn't get too stressed because that can put a strain on their heart too.
So how can cardiovascular disease be treated? So fundamentally, if you change your lifestyle, so you make healthier choices, such as exercising more. If you have a smoking habit, give up smoking. If you've gone a bit too far down the line where lifestyle changes aren't effective enough, then you have to go for a surgical procedure. And this includes, let's see what my drawing's doing today. If you've got a narrowing of your artery due to a fatty deposit on its walls, then they can put in what's called a stent, which is like a wire mesh containing a balloon which they inflate. And that helps to widen your arteries, and we call that a stent. And then coupled with surgical procedures, but slightly less hardcore, is medication. My granddad had cardiovascular disease, and I remember he was given aspirin to help thin his blood.
Right, I hope you found my video helpful, guys. These are so difficult to make, but I know you guys really like to just sit and watch the whole thing in one. So please give me a like this video if you found it helpful. It is a good incentive for me to continue my work, and don't forget to sub. I'll be back soon with another video. Bye!